So like what to put on the front of your note card, what to put on the back of your note card. So I've been debating every semester whether to just put this on Quizlet or not. But the reason I don't on purpose is because I don't want to take that motion away from you, because the act of writing note cards sometimes is all the studying you need. It's just writing the note cards, and then having the note cards is helpful for the final, so you can go through them again for the final. But sometimes just the act of writing the note card is extremely helpful. So for me, actually, once I'd write my note cards, I could go over them 2-3 times and I'd be done. So if I do the Quizlet for you and all you're doing is looking, some people don't learn well by looking. They learn by writing, actually. So there's 2 things that I'm going to tell you. Well, 3 important. The third is an important thing that you might not know yet, but I'm going to surprise you. So writing it yourself is hugely helpful. The second thing I was going to tell you was I'm not going to fill this out as we go along. I'm just going to talk. You're going to fill it out. So active listening really helps. So start writing like crazy. If it does help you to record it, you can record it and then write in and fill in the blanks later. But I do think that it's helpful that I don't just write everything down and you take a picture of it and go and study it later. So you can fill in the blanks as you go along. And then the third thing I was going to tell you that some of you might not know yet, because if you didn't ever look at that grading and participation document, you don't know this yet, but our lab practical midterm is worth 0 points. So our lab practical midterm is worth no points. But the reason I don't tell you that until today, I try and put it off to tell you as long as possible, is because I want you to study, even though it's not worth any points. Because it's a good dry run. The reason I do it this way is because it's a good dry run for the lab practical final, which is worth 100 points. Our lab practical final, we only have like 2 more or 3 more labs to add to this, and that's it for our lab practical final. That's the only things you're gonna need to know. This plus 2 or 3 more labs, and that's it. So for the final, all that time we do from now to the final is for your unknown project, which is not on the final. So if you don't study now and you wait for another month and a half to study, you'll forget all this. I promise you'll forget. So I want you to study, but the stress level can go down. Yeah.
So after we do those other 2 labs, you're saying that like, is every week between then and the final sort of self-study for the project, or is there like a structure of like week by week still?
Yeah, great question. So what's gonna happen is we'll do those couple labs, we'll do our unknown project, and we'll do a couple, maybe another lab or something like that. I can't remember what our schedule is. But how the unknown works is that you're gonna come in with a plan, and I'm gonna help you kind of design a plan a little bit ahead of time. I'll talk to you about the unknown project, what a plan kind of looks like, what to expect. And then what you're gonna come do is independently work every single day on running all of these tests on your own. So you're like, oh, today I'm gonna do a carbohydrate test. Today I'm gonna do, so also if you don't study for this, you're gonna come into the unknown project going, what should I do next? What should I do next? And you know, it's okay. We'll help you. Mostly I want you guys to help each other in your lab groups, but if you don't, even if we come over and help you a little bit, you're gonna be behind and you're gonna be confused, because it's just that you have so much content and so much to do, and you know, it gets overwhelming. You can run 20 tests, and like you will get points marked off for running 20 tests that you didn't need to run, because you're just running the gamut of tests, and you're like, well, I ran a catalase, but none of your organisms should even be catalase positive. Why would you run that? You know, like, so it will help you to understand it better. Don't stress about the unknown project right now. I will baby step you through the unknown project right before we get there, so that you'll be like, oh, okay, I get it now. But what I do want you to do now is understand the tests and what they mean. That's it. Yeah.
Will we get a list of all the possibilities?
Yes.
Okay.
You'll get a list of all the organisms it could be, you'll get a list of all the tests to run, and then you can start picking what you wanna run based on what you think your organism might be. And so you start narrowing down until you get to your own organism. You're gonna turn it in and cross your fingers and say, oh my gosh, I hope my organisms were right. If they're not right, you don't get very many points off. Mostly I'm looking for methodology, that you knew what you were doing, that you know, I'm happy to help you, but you didn't need a ton of help, you kind of got it a little bit. You're going to have questions, and that you never get points off for asking questions, but that you kind of knew what you were doing and that you can work through it. But we'll go through the unknown to death. Yeah.
Is there like a test we all start with?
So in the back of the book, I'll have you guys read, lab number 50, and he recommends, I think our author's a male, yeah, we have a male, it's a male-female combo. I hate saying he just brought me, but, in the back of in lab number 50, they recommend carbohydrate fermentation first to do all the sugars, and that does really help. But also you might not have to do all the sugars, because as we can go through, I'll show you like why you might not. Like some of the tests will be testing for the same thing, so you might not have to. But if that helps you, and I don't know if Dr. Rosemary, when we get to the unknown, you can help them a little bit with like, this is what I did.
Yeah, totally.
Awesome. Okay. Yeah.
We will get a score back though, on Tuesday. See how we did. No, maybe not, like it will be scored though.
Yes, we're gonna do it just like a test. We're going to, you're gonna fill out a Scantron. I can run the Scantrons. Oh, you know what I actually did last semester is had everybody grade each other, so we could do that as well. Or I can run them through the Scantron, and you can look at your grade right then and there and look what you got wrong and go back to that station and like, what did I do?
Do we need to bring in a scan?
No, I have Scantrons for you. All you need is a pencil. Yeah.
So the one on Tuesday will be just like the real one?
Just like the real one.
Minus the 2 or 3 extra labs.
Yes.
Will
It'll be
the-
almost identical, questions different, but almost
Same
identical.
topics, okay.
Same topics. The labs towards the end, it's a little bit more heavy towards those labs we haven't done yet.
Okay.
But not, it's still probably 75% these labs, maybe 80% these
The
labs.
setup will be the same.
I'm sorry, what?
The setup will be the same.
Setup's the
time
same,
limit for
yes.
stations.
Yes. So for the lab practical, let me run you through it really quickly because it's stressful for a lot of people. I think when some of the students got into the lab practical, it stressed them out a lot because they were like, whoa, I thought we were just taking a test. So it's kind of hands-on, but not really. But there are stations. So you're gonna walk in, put your backpack in the front because you won't need it. You just have your pencil and a scratch paper and your Scantron, and you'll start at a station and then you'll move through the stations. So there are about 35 stations, there's 50 questions, and then when you get to that station, sometimes there's a microscope and it says, is this gram-positive, is this gram-negative? Is this cocci, is this a bacilli? Sometimes it'll say like, which streak plate technique was the most accurate? What part of the microscope is this? It'll ask all those little questions at each station, and then every station you go around and mark your answer. You go through all the stations, you get 90 seconds per station. At the end, you have somewhat unlimited time. You probably only need 10-15 minutes to go back through any of the stations that were difficult that you need to look over one more time. Then we'll turn them all in. I can run them through the Scantron, and then we'll come back and you can look at them one more time, at the stations one more time, at what you got wrong, just so that you can see where you went wrong. Yeah. So the poster project, I'm hoping that we can talk about it on Thursday. Yes. So the lab practical should only take us a couple hours, so I'm hoping to talk about that on Thursday. Yeah. I need to get that going right away because I want to give you guys as much time as possible. Any other questions? So just know the lab practical, there's stations, you go station to station, you'll sit, we'll ring a little bell every 90 seconds, you'll get up, move to the next station. It's kind of like a little bit of a stressful environment, so that's why I do like to do a dry run. And then also, but this is the thing that I always debate about, is that my students generally do at about a 72, 76 average for the first go around, which is lower than I like in general, but also people tend to study less because it's not worth any points. But then when it comes to the final and everybody should have been studying and everybody knows it's for 100 points, it's still about a 76, something like that. So I want it to be higher. I'd rather an 85 average, but that takes a study, just takes time. So everybody's gotta like flip through note cards and understand the labs. But hopefully the dry run increases that last lab practical final is my goal. Hopefully your goal to get that higher score on the final. Yeah.
I think you said it's multiple choice, is the final multiple choice? as
Finals,
well?
multiple
Okay.
choice as well. And there's 5 bonus questions at the end that are worth 1 point each. And those 5 bonus questions are fill in the blank. And I don't think they're too tricky, but I can give you some examples. Sorry, go ahead, John.
Well, there will be a good amount of information from like the background sections from our lab manual. Cause
Very-
that's like a lot of where, a
Yeah.
lot of like the understanding of the labs comes from, it seems like.
Yeah, very good question. No, there's nothing really from the lab manual background. It's all just practical, like it really shouldn't be anything that's not on here.
Okay, so like, make sure you can do the labs without any help, and then you'll do pretty well.
Yeah, and like that you understood the fundamentals of the lab, because when she asks a question like that's funky about a lab, then you're like, if you understand the lab, then you're like, oh, that makes sense. But if you don't understand the lab, you're like, I don't think any of the questions are really tricky, but I've taken it and read it and done it 10,000 times, so I don't think my judgment is fair. But what did you think, Dr. Rosemary? Did you think they were tricky questions?
In hindsight, no, it's
hindsight.
not very difficult. If you're unprepared, yeah, it can be difficult, but if, you study well, it should be easy.
Awesome.
Yeah.
Awesome. Did you want to
I
say
think just having some time afterwards, like once it's graded, and then we can go back through and kind of see what we got wrong, what we had difficulty on things, and then maybe have time to ask some questions to figure out what went, why
Awesome.
we didn't get it, or,
Okay.
cause it's such a blur afterwards, after it's graded, you're like, you don't remember anything.
And it's on the Scantron. This is why I hate Scantrons.
Yes.
I can't stand Scantrons, but sometimes it can be helped. Some professors just don't have time to sit and plus.
Yeah.
But at the same time, you're like, oh, number 32 is B instead of D, and you're like, I have that's not my goal. My goal is that you have no idea why it was B and why it was D. My goal is that when it says it was the gram-negative outer membrane, that that's why the answer was gram-negative outer membrane, so that you could see, but, yeah.
So maybe not on this test, but like on the final lab
Mhm.
practicum, if we're only getting 90 seconds per station, will there be any time at the end to be able to go back and double check 1 or 2 of the ones you weren't sure about?
Yes, so usually we give about 10-15 minutes, but I will give you more time if you need it, and usually all that's needed is 10 or 15 minutes.
Sometimes you just need a second look.
Sometimes you just need to like clear your head and come back again and double check yourself, yeah.
Sorry, I don't wanna get too into it, but,
No, go ahead.
were questions asked like in a way where it would be describing something and you would kind of expect you to know that it's talking about the endoscore test? Or is it gonna explicitly say like in the endoscore test?
It'll say like, let me think if I can think of an example, like, it's not tricky, it's like, we used malachite green and safranin in what test?
Okay, yeah.
Yeah, and then it'll give you like acid fast, endospore examples.
Okay.
Yeah, hopefully it should be straightforward. And then also I don't mind asking questions, if you're like, this seems like a wild question, Like, do you mean this or do you mean this? You know, like, I like those kind of questions the best, not like, what do you mean? And I can, you know, I feel like I'm feeding answers a lot, but if you ask me like, is this what is meant by this, or is this what is meant by this, then I can help
Yeah.
more too. But I don't mind questions either. Okay, let's do it. Okay, so I'm going to talk really, really fast, so hang in there with me, and if you guys just write. Also, if you want to voice record, you're welcome to, and go back through and make sure you're feel confident with all of your answers. So on the front, it's gonna say safety, if you wanna do note cards, or if you wanna do a paper, whatever you wanna do. So biohazard, so I've seen her ask some safety questions, some biohazard questions, what belongs in what biohazard bin? If something goes wrong, what are you gonna do? So hopefully those are pretty self-explanatory. Glass goes in the sharps. If it's dry glass that's never touched anything, it can go in the glass sharps container. Nothing goes in the bag except soft stuff, so gloves, paper towels, things like that. PPE, so all our goggles, gloves, lab coats, hair back, shoes, that kind of thing. So some of the safety basics, like if you drop something, then I shouldn't just pick it up myself, like notify someone. It seems like it's all pretty straightforward as far as safety goes. Aseptic techniques, so just, you guys have been using your aseptic techniques, so you know, like, just some of your techniques, just double check that you remember that. It should be, all of those are pretty straightforward, and the multiple choice can get really silly with those, so you're like, well, just pick the non-silly answer, it makes sense. So contamination, just avoiding contamination by using your aseptic technique, stuff like that. So they're all multiple choice, it should be pretty straightforward. Microscope, so making sure you know the different pieces of your microscope, that your ocular lenses are times 10, so you'd use total magnification, multiplying it by your objective lenses for your total magnification. So if you looked at an objective lens and that's not the magnification, you have to multiply it by the ocular. Stage controls are the X and Y axis that move it around and around. The fine and coarse adjustment, remember, moves the stage up and down. The coarse adjustment moves it up and down rapidly. The fine adjustment moves it up and down slowly. And a little bit at a time. Oil microscopy, so that's when you're under 1000 magnification. Cleaning before you go back to your 400 magnification is important. Don't just drag your 400 through there. Iris diaphragm is to help with contrast, so it helps you to be able to tell the difference between the background and the organism itself. And that's the little guy underneath, and that should match your ocular lenses. If you guys remember the little dial underneath. Yeah, so that's microscopes. I'm happy to go back through that. If you guys have questions, maybe make a little note and say, can you go over this one more time at the end? Does that sound good? Okay, so gram staining. So what is the purpose of the gram stain? To dye what? Peptidoglycan cell wall. So we're looking between cell wall variation between gram positives and gram negatives. What does the cell wall look like? What is it made out of? So gram positives are what color? Purple. Gram negatives are red. And when we go through these steps, we don't just dye them in the same manner. And let's talk about that in a second. So yes, you have to heat fix. Always remember, you always have to heat fix everything that you're doing a wet mount on, otherwise the bacteria will slide right off. So step 1 was, what dye did we use?
The violet.
Crystal violet. Step 2 was the weird one.
Iodine.
The iodine is the mordant that traps the crystal violet in there. Then step 3 was alcohol to decolorize. And what are we decolorizing when we're using the alcohol?
The cell membrane.
Yes, cell, yep. So when we're decolorizing, which bacteria are we decolorizing? The gram negatives, because the alcohol will cause the pores in the outer membrane for the crystal violet to leak out. So we're washing the crystal violet out of the gram negatives. And then step 4 is safranin counterstain, perfect. Okay, so know the steps, know what the steps mean, what we're doing in each step. Okay, snot test. So snot test we did really quickly. Snot test again is we're differentiating between gram positive and gram negative. But snot test we use KOH is potassium hydroxide. That's what KOH means. And what is the KOH doing in either gram positives or gram negatives? Yep, the cell membrane. So it's lysing that cell membrane of the gram negatives and breaking apart those gram negatives. So which one is snotty?
Gram negatives.
Gram negatives are snotty, gram positives are not, because they don't have an outer membrane. Are you with me? Okay, isolation streak plate. What was the point of the isolation streak plate? It's in the name.
Isolating bacteria.
Isolate. So this is a huge, I know it sounds really dumb, but actually this is a huge part of your unknown, is being able to isolate. And it's a huge part of clinical medicine, is your patient usually doesn't just have one bacteria living there, there's multiple. So they have to isolate and figure out which bacteria are the pathogens and which ones are the commensals. So an isolation streak plate is important. So remember we did the 1, then we did the streak 2, incinerated, rotated our plate, did streak 3. So if you look on the plates and you can tell which one was done properly, that's important to be able to tell what a streak plate looks like. The purpose is to isolate, to get individual colonies. A colony is typically from one bacteria. One bacteria will grow in an individual colony. So you're trying to get from millions into one, which is very difficult. So you're pulling as you go and getting lower and lower in numbers all the way until you get to area 3. The method, was the plate and the streaks. Okay, so acid fast, the purpose of the acid fast was what?
The wax. The
Yes.
liquid wax that outside of the bacteria.
To dye the mycolic wax or mycolic acid on the outside of bacteria. What type of bacteria is the most important? What's the whole point of this test? Mycobacterium tuberculosis. So remember, a significant portion of our planet, like more than 1/3 of our entire planet has tuberculosis. A huge amount of people in the world have tuberculosis. So somebody who's trying to diagnose their patient can do it extremely cheaply and easily by doing a sputum sample and then plating it on an agar plate and then staining it. So you would take some of that Mycobacterium tuberculosis, which is scary to do. They'd have to, be really careful. But then they take it, they put it on a slide, heat fix it, and then they would dye it with what was the Mycobacterium tuberculosis stain. It's close to red. It's hot pink. Carbol fusion. So carbol fusion always reminds me of the word fuchsia because it's hot pink. So those Mycobacterium that are tuberculosis causing organisms are hot pink. So the stain is carbol fusion. Then we used a counter stain to be able to detect, because we had 2 different organisms, because generally you will have more than one type of organism. So it's always good to do a counter stain to see what else is in there, what else is going on. So we used a counter stain for a different organism. Do you guys remember what organism we looked at? This is not really that important, but we looked at Staph aureus, I think.
Yeah.
So yeah, we used Staph aureus, and then what was the counter stain that we used? We used, what was it? Methyl? No, we used, methylene blue. So we used a blue stain. So this stain, if you looked at a slide that was hot pink and blue, the hot pink are gonna be the Mycobacterium. Mycobacterium, on the back of our note card, if you remember from our Mycobacterium tuberculosis note card, we weren't sure what the peptidoglycan was. They are thought to be gram positive, but we don't really stain them gram stain. So we stain them with the Carbol Fusion, they're hot pink. We use methylene blue as a counter stain. That was the Staphylococcus aureus. So if you see a hot pink and blue, Mycobacterium looks like this. They're long bacilli. Yeah.
So the counter stain, the purpose of it is just to see what else we have on our plate.
Yeah, and just so, it was to give you kind of a mix and to see, and then also what they like to do a lot in the lab is to give you like mystery patient. Does your patient have tuberculosis or not? So then you know like, oh, if there was no Carbol Fusion and it was just Staph aureus, then you go, oh, it's the staph infection. Or no, it's a mycolic acid, it's a mycobacterium infection. Yeah.
Is it that tuberculosis is so like omnipresent that you don't have to worry about the possibility of it being a different mycolic wax-based bacteria if you're doing this kind of stain, and you can just assume that if the test is positive that it is tuberculosis?
Yes, you would do something confirmatory, but yes,
Okay.
yes, awesome.
This would be one of the bacteria that you, if this came back positive, you would have a good idea of what your bacteria is.
Yes, then it would be, yes, that's what it's used for, is the Carbol Fusion is used for. Awesome, yeah. Okay, cool, then it has that mycolic wax on the outside.
Sorry, yeah, so
Yeah, yeah.
like in the actual setting, would you more so use that test once you already thought it was tuberculosis, or would you do this first, like without that?
Well, it probably depends, like if you're in a developing country and your patient has been coughing for 6 months, then you did an acid fast stain, then you do an acid fast stain, but I mean, yeah, they could do it like they don't have any symptoms, but the people in their house, you could test the people in their house and say like, do they have it yet or not?
Okay.
But then generally we have more sophisticated methods. But if you're in a developing country, this is a really cheap, rapid way to do it. Yeah. Awesome. Okay. So endospores. So what was the purpose? Figure out if your organism is an endospore forming organism. So what was our endospore forming organism? Yeah, Clostridioides difficile does form endospores. That's a very scary bacterial infection. What was the one we used in the lab? Bacillus cereus is an endospore forming gram positive bacilli. Bacillus is in the name, it's a bacilli, but it's a gram positive rod versus a gram negative rod. It's a gram positive rod, and they form endospores. And so we're dying the endospores versus the endospore versus what is the other cell type? It's vegetative. Awesome. Yeah.
What was the new versus the old Bacillus cereus that we did in lab?
Yeah.
I
So
just didn't understand the new versus old part.
awesome. So the new part, we're looking at probably mostly what type of cells? Vegetative. So we're looking at mostly vegetative cells because that's a new culture. Where if you let this plate sit for a long time, then it runs out of nutrients, it's starting to get old, they're forming more endospores. So that's why we did a new versus old culture. The old culture probably had more endospores in it, the new one probably had more vegetative cells in it. Does that make sense? Why? Yeah, because it starts running out of nutrients and forming endospores. Yeah.
Sorry, I must have just missed it, but what was the purpose?
So the purpose was to figure out if your infection was from an endospore forming organism, or just to detect if there's endospores. And then so Bacillus cereus, we used, what was our endospore stain? Do you guys remember the procedure for this one?
Would you heat fix it?
Yeah.
We heat fixed it, but then what else did we have to do aside from the heat fixing? Another method of, we did the steam bath. So we did the steam bath to break open the endospore outer shell to hopefully dye those endospores, to get them, I won't tell you, what is it called? The dye, do you guys remember the dye? Malachite green. So the malachite green to bubble and steam it to get to the malachite green deep into the endospore layers. So that's malachite green, and then the counter stain was what? Saffronin? Saffronin. Yeah, saffronin. So we had bright green endospores and reddish, pinkish counter stain for vegetative cells. They are bacillus, it's in the name Bacillus cereus. They're rod shaped, and they are gram-positives, because most gram-positives are most endospore-forming organisms are gram-positive. Okay. Blood agar. So what was the purpose of our blood agar? Yes, to compare organisms to look at hemolysis. What is a hemolysin? Yes, it's an exotoxin. What type of exotoxin? 1, 2, or 3? So it's a type 2 exotoxin. These, to look at a bacteria, if it spits out this hemolytic type 2 exotoxin, and what are we mostly looking for when we do a blood agar test? Do you guys remember what we're looking for? Well, this is kind of a funky question because we don't use this organism in the lab because it's too virulent to use in the lab. But what would you use it for? A lot of times, what they're looking for when they use it in a clinical lab. Strep pyogenes. So they're looking for a group A strep pyogenes infection, strep throat. So that's why they'll use blood agar in a clinical lab, is because they're looking for strep throat. What organism did we use that's also a beta hemolytic? Staph aureus is also a beta hemolytic. So I'll just write that up here too so you guys don't forget our organisms that we used. So one was Staph aureus, which was a what? Beta. Beta, which was the complete hemolysis. And then what else did we use? Do you guys remember? Epidermitis. So epidermitis, so sometimes if you don't recognize an organism when it says it on the exam, try to see if you can identify what the organism is based on the name of the organism. So what does epidermitis remind you of? Skin. So it's an epidermal infection, and actually it's a commensal. They live on us and around us all the time. What was epidermitis? Was it alpha, beta, or gamma? Gamma. So gamma was no hemolysis, right? And then what was our third one? Faecalis. So afacalis, she won't ask you about that, was an alpha. So that's partial hemolysis, no hemolysis, complete hemolysis. What else do I want to say about that? It's a type 2 hemolytic. Is it selective? What does everybody really quickly think in your mind? Selective versus differential. What does selective mean?
Send the employees home.
Yes, awesome. Send the employees home. We're gonna select. Or differential. So what does differential mean? They make the employees wear different shirts. Awesome. So great. I'm glad some of my dumb analogies are working.
Like you should get candy for that.
Yes. Oh, I agree. 100%. You totally get candy for that. There's all kinds of good stuff. You gotta put it on. Okay. So is blood agar selective? Are we sending anybody home, or is it differential? Are we coloring between the organisms? It's differential. It's not selective because you could tell the difference between the organisms. Nobody died. Nobody got sent home. Okay. So EMB, what does EMB stand for? Eosine methylene blue. Eosine methylene blue. What was the whole purpose? This would be one of the ones that maybe the background would help you on this one. What was the purpose of the EMB lab? Yes. What did you say, Dr. Ferguson? I'm sorry, there was. Yes, definitely lactose fermentation, but why would they use this? They wouldn't necessarily use this in a clinical setting. They use this where, Yes, fecal coliforms, awesome. So EMB is really to detect fecal coliforms. Is there feces in the water or not? So the purpose is to detect fecal coliforms. If there are organisms that typically live in the colon in the water, then there's feces in the water. So organism 1, what was one organism we looked at? E. coli. And what was another organism we looked at? S. aureus. What was another one we looked at? Pseudomonas aeruginosa. We did?
I don't actually know if we ended up
No,
looking
I
at
don't-
this.
yeah,
I think we did Enterobacter.
Enterobacter?
Yeah.
Enterobacter aerogenes. Okay, so is this selective or differential?
Both.
Both. And why was it selective?
Selective for gram-negative bacteria.
Yes, so selective for gram-negative,
And it was differential for fermentation.
and it was differential based on fermentation of what? Like Dr. Ferguson said, lactose. So there's lactose in the medium. So E. coli was a rapid lactose fermenter. Enterobacter was slow, is that right? Or aureus was slow? Well, no, aureus, what happened with aureus? It didn't grow, it died, right? It died. So it died because, why?
It was gram positive.
Because it's gram positive, so we don't know if it ferments lactose or not at this point, right? So what about Enterobacter aerogenes? Slow. Okay, so we'll just say slow, rapid. And so this one was neon green, this one was purple, this one died. Good? Do you have a question about that, Dr. Winters? You feel good about that? You want me to go over it one more time?
No, I think it was, they said pink means slow.
Oh, yeah, no, it's pink, you're right, it is pink, yes, awesome, good point. Yeah, because purple is the color already in the medium, so it does look, it's more pink than the actual medium is purple, good point.
Sorry, it's selected for lactose fermentation.
It's selective because the people it sent home were the aureus, so it sent home the gram positives.
Oh, sorry, differential.
Oh, differential because the lactose fermentation, yeah. So it's creating an acid. This one creates lots of acid, this one creates a little bit of acid. Yeah.
So we would say it's selective for gram-negative though, correct? That sends home the gram-positive.
Yes, that is a great point. Some people have a hard time with that terminology. It's selective for gram negatives. It selects for, yeah. Yeah, you know, I messed that up because mannitol, let's do mannitol now, but I put it in the wrong space. So let's do it here because I forgot about it and it didn't go with my order. But here's mannitol salt, yeah.
We had a question earlier, why are mannitol salt, the blood, and the EMD grouped together?
It's kind of like our selective differential topic. Yeah. We just clumped them together because it's like, is this selective differential? Yeah. Good point. Okay, so here's our mannitol salt agar, MSA. That's what it stands for. What is the purpose of our MSA?
Differentiate between halophiles and non-halophiles.
Yeah, so halophiles and non-halophiles, usually.
It selects for halophiles.
Yeah, so all of those things, so we're looking more at like organisms that cause skin infections because we're looking at that organisms that can grow on salty media. So we're looking at organisms that are skin friendly or non-skin friendly. So the purpose is to differentiate selective, we'll talk about that in a second. So what was our organism 1? Aureus is a huge skin issue. What else? E. coli, we looked at for a reason, we'll just talk about it. And then what was the last one? Epidermitis, which tells us something, right? That it grows on the skin. Okay, so was this selective or differential?
Both, I think.
It's both selective and it's differential. Who'd we send home?
The non-halophiles.
Yep, we sent home E. coli because it's a non-halophile. It can't survive in salt. That outer membrane makes it to where that it's not happy in a salty environment. So here Staph aureus was survived, Epidermidis survived, and then it's differential because what are they doing? pH based on, what is it fermenting? Sugar fermentation of mannitol. Mannitol is the sugar. This one is a sugar producing, and this one was yellow. This one was pink. This one was dead. Okay. So that one is selective and differential to differentiate between halophiles, non-halophiles, mannitol fermenters, non-fermenters. Usually we look at it when we're looking at skin infections. Okay. Now plate count. So plate count, I know was a beast. Did you guys look at Dr. Welsh's video? Did he post a video? I think you might have. Yeah. So Dr. Welsh is a great resource also for this test. He's, I think, maybe the only one I know of that has ever gotten 100% on both tests. I don't know anybody else that has, but he studies a lot. But plate count is what was the purpose of the plate count?
Determine if the milk was safe to drink raw.
Yes. Is milk safe to drink? Raw milk safe to drink, or any food item at all safe to drink or eat? So what did we do with what is cereal dilution? So I mean it's kind of in the name. You're just cereal diluting. Sorry, funky question. So what you're doing is you're mathematically given these values of dilution that you have to follow according to the U.S. government. You cannot plate raw milk on a plate, otherwise it'd be all bacteria. And then they'd be like, well, it's not safe. Well, it is safe, just not at such high concentrations. So you have to dilute it, and the government tells you this is how you do it. You measure the part over the whole. So remember, our dilutions were like if you had a 1 milliliter over 10 total milliliters in your bottle, then you'd have a 10 to the -1 dilution. And then if you diluted another from that dilution, then you multiply those two dilutions. So if you have another 1 over 10, 10 to the -1 times 10 to the -1 is 10 to the -2. That's your final dilution for that. Then you keep going, then you keep going. So you're serial diluting as you go along and along, and then you have to measure the aliquot. How much are you putting onto the agar plate? They want to know that as well. They want to know how much you're diluting, and they want to know how much you put on the plate. If you put a gallon on an agar plate, is that valid? Is that you'd have so many bacteria regardless of how many are in the solution. So you have to know, you have to give them that number. So you're factoring in the aliquot. Usually it's 1 milliliter, but not always. So you have to watch for that number. And then remember, this always helps to look at CFU per milliliter. CFU stands for what? Colony forming units per milliliter. So CFUs, and then you look at the dilution times the aliquot. And so then you'd have some number per milliliter. What if those CFUs are, if you had 17 colony forming units? Awesome, it's not valid because you can't count anything under 30. If it's 300, it's, yes, but 300 officially, if it's anything over 300, so you have to be within that 30 to 300 range. Then, then you're counting your colonies, you count your colonies and you put this number, your dilution number, you put this number here in the denominator and you have CFUs per milliliter. So this is a great one to watch Dr. Welsh's video. I can help walk you through this again after class today or anytime. And then, what you wanna be able to do is make sure that if she says you have 210 colonies and your final dilution was 10 to the -5 and you aliquoted 1 milliliter, how many CFUs per mil? So be able to calculate that. If she says put 810 into scientific notation of liters to milliliters, so being able to do that unit conversion. If you guys want an extra little go around with that, I'm happy to do that. Here are the pipettes. So always with the pipette, start by looking at the top. The top, these are always in microliters, microliters, microliters. 1,000 microliters is how many milliliters? 1. So remember we had that little cheat sheet on the side of our lab notebook. Our little cheat sheet said 10. Let's do that after, because we do have a little cheat sheet, but I want to keep going. So we had that little cheat sheet in the side of the whiteboard that told us how many microliters are in a milliliter. So let's go over that if you need more help with that. But you're going to start with the pipette. Look at this. This is P1000. That means there's 1,000 microliters. This is capable of aliquoting. And so if it has capable of doing 1,000 microliters, that's 1 milliliter. It can only do 1 mil max. I always also start by doing this. If it goes down and a lot, down far and up far, it's almost to its max. If it barely, you can and it barely clicks up and down, it's closer to its minimum, which on this one is 200. Then you have to look at the little dials here, and I can bring up that slide again if that helps you. When you have a P1000, what this number means, what this number means, what this number means. So you might have to just get one note card and memorize that of comparing a P100, P10, P20, and have that on a note card. You can even print that slide and put it on the back of a note card so you remember which places. But really, you should be able to, from your A, B, C, D, E option, guess which one it is based on the maximum and minimum value and pushing the top. You should be pretty close to is it about a milliliter or is it barely clicking up and it's 10 milliliters? This one is a P10. If it's going all the way down and all the way up, it's close to 10 milliliters. What on my options are 10 milliliters? So start there and see if that helps.
Right?
I'm sorry, microliters. Yes, thank you. Always catch me. Microliters, so 10 microliters would be when it's almost all the way up and down. If it barely clicks, it's closer to 1 microliter. Yeah? Okay. So look at these. Don't tell anybody I did that. So look at these before you go play around with them. Okay. So the plate count tells us if it's safe, if it's not safe. That one, I don't want you to tell you not to study it, but there's maybe 3 questions on it. So if you're struggling, make an appointment between now and the final with me, Dr. Welsh, Dr. Rosevear, to sit down and go over it one more time, but try not to stress about it if you feel super lost. Focus on other things. Get all of these 100%. If you miss 3 questions, that's okay. Okay, gelatin. So the purpose of the gelatin test was to assess for what enzyme? Gelatinase. That's easy, right? So the purpose is to assess if your organism spits out the gelatinase enzyme that breaks down gelatin. Gelatin is basically collagen that's in all of our connective tissue, so it's going to break down connective tissue. What is the method? What's funky about the gelatinase test? What do we have to do that's special?
Heat it up and then refrigerate it.
Yes, we have to put it in the refrigerator because it's liquid no matter what in the incubator. If we put it in the refrigerator and it's still liquid, it made the gelatinase enzyme. If we put it in the refrigerator and it's solid, then it doesn't have the gelatinase enzyme. Okay, carbohydrate fermentation. Oh, I'm sorry, no, let's keep going. Enzyme is gelatinase. What an enzyme and substrate, what that means, an enzyme, remember, is just a protein within the bacteria. So we'll draw a big bacterium. We'll draw a big giant enzyme, even though this is definitely not to scale. here's an enzyme. What substrate means is it's the target of this enzyme. So that's what substrate means. So the enzyme would be gelatinase, the substrate would be gelatin. That's what gelatinase cuts. A positive looks watery, a negative looks solid. Okay, carbohydrate fermentation. So this is also called phenol, what is it called? Is it phenol red sugar broth? Phenol red sugar broth, it's also called. So carbohydrate fermentation test is also called phenol red sugar broth, which also tells us something about this test. So phenol red sugar broth were our 3 sugars, which were glucose, sucrose, and lactose. So here, if we have one that's lactose, a lactose fermenter, what color was it? What color was the carbohydrate fermentation? Remember, we had the 3 tubes that had the 3 different color tops. Yellow. It's yellow if there's an acid in there. If it's fermenting lactose, it has the lactase enzyme, there's an acid in there. So if there's an acid in there and it ferments lactose, what else are we looking at lactose? What other tests are we looking at in lactose? Yes, but what other tests did we test for that? EMB. So here we know E. coli is rapid, Enterobacter-Rogenes, yes, it ferments lactose also. Both of those would be lactose fermenters, so just so you know, we kind of cross them. So this one I put kind of simple because hopefully it helps. Purpose was to assess sugar fermentation. Does it ferment? Is it a fermenting organism? If it's a fermenting organism, what do we call it? Facultative. It wouldn't be anaerobic because they won't grow in our lab. It needs no oxygen. It would be a facultative. It wouldn't be aerobic because aerobic organisms do not ferment. They don't create acid byproducts. So it created an acid byproduct. That's what the phenol red is. The phenol red is detects acids. It detected an acid, so this, if it has an acid, it's going to be yellow. What was one more thing we tested for? Gas production. So does it produce CO2 or not? So just because it produces CO2, so some of these organisms will produce CO2 and some won't. So it's just a secondary test. Does it produce CO2? Does that help us to determine what organism it is? That's all that helps us.
Would CO2 be a differential part of the test?
Yes, yes. Because some are like, for example, Staph aureus is just acid, and no gas. It doesn't produce gas. But E. coli is acid, gas. Can't remember off the top of my head. But they do produce gas, whereas Staph aureus doesn't. But they both ferment. Okay, so here's our fermentation chart. We're looking at, is it an acid, or acid? Okay. So remember, acid, gas is the Durham tube. Let's move on to catalase. So catalase, so just really quickly in your head, we had 2 very similar tests that we did at the exact same time. The only reason they're similar, well, they are similar for a couple reasons, but we had catalase and what? Oxidase. Catalase is looking for what? So we're looking for this, does it have this enzyme, and what did we do to see if it has this enzyme? See if you can remember without looking at your notes. Hydrogen peroxide bubbles. So this one has bubbles. So what we're looking for is this enzyme that breaks up hydrogen peroxide. So we're looking, does our organism have the catalase enzyme? Catalase enzyme usually tells us that it's at least has some sort of electron transport chain. So yes, if it has the catalase, it probably has an electron transport chain, meaning that it's aerobic or facultative. It will, the catalase enzyme breaks up the H2O2 and turns it to water and oxygen because hydrogen peroxide is toxic. Our bodies will make hydrogen peroxide on accident. So will bacteria. So the purpose, does your organism have it or not? Method is H2O2, bubbly, and then the enzyme is catalase, the substrate is hydrogen peroxide. Does that make sense? Yeah, what the substrate means? Positive, bubbly, negative, not. Okay, moving on. Here we go. Okay, oxidase, the purpose of the oxidase test. So really quickly, what does the oxidase test look like? The little strip. When you say the word oxidase, you always say what? It comes cytochrome C oxidase, cytochrome C oxidase. We don't really say oxidase by itself anymore. It helps us remember cytochrome C oxidase. Cytochrome C oxidase is an enzyme that helps us detect cytochrome C. So the purpose is to look for that cytochrome C. Remember, cytochromes are those proteins along the cytoplasmic membrane of bacteria. And the cytochromes are telling us that they have an electron transport chain. The methods were that little strip, and it turned bright purple, bright blue within 10-15 seconds. The enzyme is oxidase, the substrate is the cytochrome C. Positive, purple, negative, nothing. Good? Okay. We did MSA already, urease. So urease, the purpose of the urease test is, does your organism have the urease enzyme? So enzyme is the urease, the substrate is what? Urea. And if they have the urease enzyme, the enzyme will break up urea and form what? Ammonia. Ammonia, does it have a high pH or a low pH? High pH, that means it's basic or alkaline, and it is, creates ammonia, which is scary when it's in our bodies because our bodies don't like ammonia. Enzyme substrate, positive was what color? Pink. Negative was the same color it is, the same control, like kind of yellowy, pinky, so something like that. But one thing about urease, what did I want to say about that? Enzyme, urease, substrate. I can't remember. Hopefully it'll come back. Okay, so yes, that's the point of the urease test. So IMBIC, so now we're starting on IMBIC, and then we're done. IMBIC, we have SIM, MR, VP, citrate. So I stands for indole, M stands for MR, V stands for VP, I is just a placeholder, C stands for citrate. Not catalase, not anything else. It stands for citrate. So citrate, I'll show you how I remember that. Okay, so SIM, S is for, to look for, it does your organism produce what? Hydrogen sulfide, the black hydrogen sulfide. So what it's doing, actually we didn't really go over what it's actually doing, but what the hydrogen sulfide is, it's a byproduct of an enzyme that will break up some amino acids. So just take it on face value. It's work from an enzyme. If the organism has this enzyme that will break up an amino acid, it'll turn it black. Keep it simple. Positive means black, negative means not black. Here, purpose of I is called indole. This one is a little tricky because indole is a funky word that doesn't seem like it has anything to do with it, but it does. The purpose of the indole test is to see if it has what enzymes. Anybody remember? Without looking? Yeah, no, that's okay. So it has tryptophanase, which breaks up tryptophan. So the enzyme is tryptophanase, the substrate is tryptophan. When it breaks up tryptophan, when the enzyme cuts tryptophan, it makes indole. That's why it's called the indole test. So that's the resulting byproduct of the enzyme is called indole. That's why it's called that. The reagent, what reagent did we use? Kovacs. So however you need to memorize that, like make some sort of mnemonic, make some sort of little story about IMVIC and the different things that you use for the different tests that you use for IMVIC. Because you will be asked lots of questions on IMVIC for this one and the final. So this, the reagent was Kovacs reagent, and you put those drops on the top of your sim tube. So you put the drops on the top of the sim tube, and that Kovacs reagent turns it what color if it's positive? Red if it's positive, not red if it's not positive. Okay, our last one is M, which stands for motility, which just means movement. Is your bacteria able to move on its own or not? Does it have a flagella? That means motility. Is the flagella moving? So if it's hydrogen sulfide negative, but it's motile, it will have spread throughout the tube, but just look clear. Like it'll just look like bacteria spread out throughout the tube. If it's motility positive and hydrogen sulfide positive, the whole tube will be black because it's moving throughout the tube. Okay. So now moving on to MR. So MR and VP kind of go together. If these two were alone, that would be great. Oh, that was what I was going to tell you, was the reason why, even though we didn't really talk about that a ton, but the reason why we use IMVIC as a test and the reason why we ever put those together is because this really helps us differentiate between different enterics. So we look at different bacteria that live in the intestines that tells us if it's an enteric or not. So if it's, we can differentiate between organisms in the colon by using IMVIC is the point. So we're looking at MR and VP. So MR and VP, we're looking at fermentation. Does our organism ferment and create an acid? Does our organism ferment and create a non-acid? We had one non-acid example, which was this one right here, was the 2,3-butanodiol. But the reason it's tricky is because this is the substrate of the enzyme is acetoin. So acetoin, it's going to be able to detect for VP, the VP reagents are actually, I should have said enzyme, but the VP reagents are looking for acetoin. That's how they will change color if there's acetoin present. And that is on the way down to 2,3-butanodiol on that little chart. Does everybody see that? Here, 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 and here. Okay. So here is if it creates an acid. So does it create here, here, or here? Does it create an acid? So generally, if it's MR positive, it's VP negative. And if it's VP negative, it's MR positive. So that's broadly, if it creates a neutral end product, it's neutral. If it creates anything that's not neutral, it'll be acidic. Okay. So here, our last one is citrate. So the purpose of the citrate test to see if our organism will produce, it is not citrate, produce citrate from citrase, from citric acid to citrate. Can't remember. Don't quote me. So the purpose is to see if it can use citric acid as a carbon source. Can it use citric acid? So remember in our Krebs cycle, citric acid was a product of our Krebs cycle. Can it use citric acid as a carbon source or not? So the enzyme is citrase. The substrate, I believe, is citric acid. It must be the substrate is citric acid, and then citrate is the result of citric acid, byproduct of something like that. Don't quote me. Okay, so the reagent, we don't have a reagent for that one actually. That one is just, we remember it was the bright blue one. And so positive was blue and negative was green, right? positive was the bright blue. So citrate reminds us of like citrus. So if you think of like a citrus fruit, like cocktail with citrus fruit in it, and it's bright blue. Citrate is the bright blue. And then also what they'll do in the test is what we set up is if we're looking if it's positive citrate or not, then there'll always be a control there. So you could tell the one that's a control and tell the one that's been inoculated. And so if they match, it's negative. If they don't match, it's positive. One thing that's funky that I want you to know, for this one as well, that this one is alkaline byproduct. So this one is alkaline. Oh, I shouldn't use an A. Let's use a base. This is basic. Looking for a base. What other ones are looking for a base? BP, not quite, because that one's neutral. Urease is a base. Anything else? Just those two, that's helpful. Okay, so acid, fast, endosort, blood, EMB. EMB is looking for an acid. Catalase is looking for not an acid. Carbohydrate fermentation is looking for an acid. This one is looking for an acid. This one's looking for a neutral. Anything else? No, I'm not sure actually. Okay, sound good? So color change doesn't always mean acid, it can also mean basic, depending on the test. Everybody all right? A little rough, gonna go home and start sneezing in the shower? Yes. Okay, well, let me know if you guys have any questions between now and Tuesday. Don't not study, please, please, please. Study, but also we have our quiz on Tuesday, so study for your quiz. There's a lot going on.
You need to know the organisms associated with each test.
Ooh, I should have gone over that, but I'll go over that on Tuesday. Is just how to recognize some of the names of the organisms. So like Staph aureus, we know what that looks like in a microscope. E. coli, hopefully we know what that looks like in the microscope. Gram-positive cocci that are in the clumps. Gram-negative bacilli. Bacillus cereus is bacillus. It's a bacilli, but also it's gram-positive. It's a gram-positive rod, not a gram-negative rod, but you'll never have to guess that. You just look at the microscope. But it's gram-positive, we know, because it's an endospore former. Mycobacterium tuberculosis, we know what that looks like in the microscope because of the mycobacterium test. Because it's carbon fusion, it's fuchsia, yeah, and that's a rod. Staphylococcus is same as staphylococcus here, so it would look the same under the microscope. Enterococcus is an entero, it's intestinal cocci. Pseudomonas aeruginosa is a gram-negative rod. Salmonella typhimurium is a gram-negative rod.
Are there any others you want us to learn for the?
Enteric, is this one an enteric?
No.
No, that one is a long skin. Enteric, that's salmonella from chicken. Enteric. Enteric, and then this one can cause enteric infections as well. This one too, and this one can cause enteric infections as well. Remember, aureus can cause everything. C. diff is enteric.
Okay.
Yes.
Were there any other things you wanted to add to that list? We were talking about looking for.
Looking for what?
You said that there were some other organisms you wanted to add to that list.
I don't think there's very many more. I don't think she'll ask about A. faecalis. I don't think she'll ask about Enterococcus faecalis either, but it says entero in there, so I just threw it up there because it tells you it's intestinal.
Okay.
I can't remember any of the other ones that we worked on. Salmonella typhimurium, we did a lot. We switched them up this semester because we usually use Pseudomonas aeruginosa a lot, but because of the safety protocols, they don't always use it anymore.
Yeah, that's what
Okay.
she said, general.
Sounds good. Thank
Good?
you.
Ready to go?